Hi everyone. In this tutorial, we're going to draw a diagram that represents what happens in DNA replication. So we've already gone over what semi-conservative replication is, and so now we're looking at how exactly is that semi-conservative replication done and what kind of enzymes are involved and that kind of thing. And similar to cellular respiration with all those diagrams, I think it is the best and the clearest to draw it through together. And so in the diagram, in the handout that I posted, you have an explanation at the bottom and then a big square, big rectangle where we will draw what's going on. And hopefully between those two things, it's really clear how this works. So first thing that we're going to do, I apologize if we go in and out of focus here. This document camera is a little bit picky, a little bit temperamental. So first thing we're going to do, like I said, is to draw our double helix and draw it already open. And so we'll start over here by, here's my one strand, and I'm going to have it open around here. Okay, and the other strand will be like this. And I really, really recommend that you draw exactly what I'm drawing because when we start talking about directionality of each strand, you might get really confused if you're drawing it slightly different than I am. So try and do it as similar to my diagram. You might even end up doing a better diagram than me, but at least have the same amount of loops at the beginning here. So I'm just going to draw lines like this just to kind of show what we know to look like DNA, the typical kind of cartoon drawing. And then over here on the right, we've shown that the double helix has split apart. And so at the bottom, like I said, you have a description of what's going on. So we're going to go back and forth, talk about in words what's happening, and then hopefully be able to show it in the diagram. So the first thing, gyrase, as soon as you see A's, you know that it is an enzyme, most likely, and it relieves any tension from the unwinding of the double helix. It's kind of like when you have thread, if you ever do needlepoint or use embroidery thread, there's actually multiple threads within each thread. And so sometimes you have to unwind them. And if you've ever tried to do that, there's so much tension that builds up in the opposite end of the thread from where you're opening it that sometimes you have to end up ripping it if you're not careful. And so same thing can happen with double helix, DNA, and obviously you don't want it to be damaged. So gyrase relieves any tension. Another name for gyrase that sometimes you see, topoisomerase, is another word for it. So it relieves tension from the unwinding of the double helix. And the way that we're going to draw that is pretty simple. We'll just make sure we draw it a little bit upstream from the replication fork. That's what this is called, replication fork. It kind of forks out, kind of like a fork in the road. So they call it a replication fork. So we'll draw gyrase a little bit ahead of that, relieving any tension. Okay. The second bullet, the second point, says helicase, another enzyme, breaks the hydrogen bonds and unwinds the strand. And so in previous lessons, you would have learned that all the bases, A, C, G, T, they're all bonded. So A's and T's bond together and C's and G's bond together. And it's hydrogen bonds that keeps them connected together. And so hydrogen bonds are not as strong as something like a covalent bond. So they can be broken and then they can be connected together again, a little bit like a magnet almost. So helicase is what's actually breaking the bonds. So it needs to be more like here. If you want to add color to this diagram, probably a good idea. I will eventually, but each enzyme could even be a different color. Okay, so there's helicase. Next one, number 3, says single-stranded binding proteins stick to the newly exposed template strands to prevent it from winding up again. And so what we're going to do before we draw in the single-stranded binding proteins, we're just going to show, just by dashes, these are representing bases, just one base, because we've already separated them from the other bases. So it looks like half of a ladder, basically. Do them as equally distanced from each other as we can get it, just by eyeballing it. So you've split the ladder in half, and so you don't want this thing to wind back up again because the whole point is to make a new strand. This is your template strand, so it's already there. Sometimes we called it a parent strand. Maybe we can write that actually, template strand. Sometimes we called it parent. It's like the original strand. we don't want it to wind back up. We don't want hydrogen bonds to be made again. And so you actually have, just to make sure it doesn't happen when they're in close proximity, you have these proteins. They're not really enzymes. They're just proteins that sit on the DNA and stop it from winding back up. So these, I'm just going to call them SSBs, and that you can write here for short. And this, we'll use our terms, this is called the replication fork, kind of like a fork in the road. Okay, then the primase, something called primase is super important. This is step 4, lays down RNA primers that DNA polymerase 3 needs to use as a starting point. So let's start with the word primer. Whenever you use that word, if you're painting, for example, primer is done first so that the next coat of paint, the color or whatever you're using, will stick better and will be brighter. And so the word primer just means you're kind of getting ready for something else to happen. So if you think of that, it'll help you figure out why primers are needed, what they are. So they're made of RNA. That means that they're made of ribonucleotides. And so they're not deoxyribonucleotides. So eventually we're going to have to switch them out for deoxyribonucleotides. So for now, they're just kind of there to get the reaction to be able to happen, to get the process going, and we'll have to switch them out later. So primase is the enzyme that does it, and it's laying down primers. So we're going to start on the top strand, and we are going to do that because it's a little kind of the easier strand to deal with, and you'll see why. So I'm going to do primers in pink because we want to differentiate between RNA and DNA. So my RNA primers are going to be pink. So I'm going to go up here and I'm going to start laying a couple primers. We'll just do a couple like this. Okay, so it's the other half of the double helix. And this is an RNA primer. And we can draw kind of a circle over top that will represent the fact that primase is what does that. Okay, so this is primase laying down primers that are needed to create the rest of the strand. So after that is done, you have DNA polymerase 3 is what step 4 says. So DNA polymerase 3, like any enzyme, is really specific, and it needs a starting point. It needs a primer to be laid down. And so that's why laying it down first is needed. Otherwise, you wouldn't get a new strand out of it. So we're going to draw in a different color. I'm going to use green for my DNA. We'll draw a few more. So this has all been laid down by DNA polymerase 3. And it's working in this direction. So here's our DNA polymerase 3. And the word polymerase also tells you what the enzyme does, because you see the word polymer. Polymer means putting lots of little pieces together, and ase is an enzyme, and put DNA with that. So what is this enzyme doing? It is taking nucleotides, deoxyribonucleotides, and putting them together to make DNA. So all your clues for what this thing does are right in the name. Okay, so the overall direction that it's making this strand in is that way. Okay. Step number 5 says DNA polymerase 3 adds deoxyribonucleoside triphosphates. So essentially new DNA. It's in triphosphate form. It's not really necessary that you know that, actually. Just know that it's laying down new DNA or daughter DNA. Using the template strand as a guide, and that means, as a guide means it links up adenines with thymines in a double bond, and cytosines and guanines linked together in a triple bond. And this new strand is built 5' to 3'. So what's going on there? Well, we know that DNA has directionality to it. So on the 5' end, there's the phosphate. If we were to zoom in and see details of every single atom, the 5' end has a phosphate and the 3' end has a hydroxyl hanging off the end. And if you're not sure about that, you should go have a look at a diagram and let me know if you're not sure about that. So 3' and a 5'. And so let's look at one of the strands. I'm going to start here, and you should start at exactly that point. Let's say that a phosphate is what's hanging off that end. So it's the 5' end. Okay, the 5 versus 3s depending on what carbon it's hanging off of. So if we follow that strand along like this, we'll find that it ends up here. And so that end must be the 3' end because there's a 3' and a 5'. That is really important that we get that right, because if number 5 is going to make sense, then the other strand, so this is the template strand in the pencil, the colored strand is the new strand, and it's the opposite of the template strand. So that means that this is 5' and this is 3' because they're anti-parallel. And hopefully that's a familiar term as well. Okay, so step 5 says that DNA polymerase 3 uses the template strand as a guide, and the new strand, or the daughter strand, is built in the 5' to 3' direction. So we can see that the phrasing of that, it's built in the 5' to 3' direction. DNA polymerase 3 has to work like that, builds in the 5' to 3' direction. It does not go in both. So that's why knowing what's 5 and what's 3 becomes really important. So this is all done, and if we keep opening the replication fork, the DNA polymerase 3 will keep adding, and it just, we needed is the primer at the beginning, and it will keep going. And so as a result of this is why this is the easier strand. You'll see that in the next part of the video, in the next strand at the bottom, it's not exactly the same story. So this is called the leading strand. And in the next video, we'll go over what happens in the bottom, how it's slightly different.